Alright guys, here's a quick review video for tomorrow's test. First of all, a little bit about the microscope. In this structure, this diagram, structure 9, the eyepiece has a lens inside it. It's also called the ocular. That eyepiece, pretty much in all microscopes, magnifies things 10 times or 10x. Another piece that I want you to be familiar with is this piece right here. That entire piece is called the nose piece. And it can be rotated to put different lenses into place over the stage. These actual lenses that can be rotated into place are called the objectives. In most microscopes, most compound light microscopes, the smallest one is a 4x objective, the middle one is a 10x objective, and the longer one is the 40x objective. Let's say we had the 40x objective in place over the slide. So we have a 40x lens here. We also have the 10x objective in the eyepiece. That will give us 10 times 40, or 400x total magnification for the object. It'll look 400 times bigger to you through that scope than it really is. A couple of other things that we need to be familiar with. We'll do a little erasing here. Underneath the stage, and by the way, it's a black flat object, that's the stage. There are clips on there to hold your slide on. Those are called stage clips. But just underneath the stage is a little lever that controls an opening. That lever and the opening are called the diaphragm, and they control how much light makes it up through the stage. The final two pieces of the scope, or actually not the final two, but another couple I want to point out to you are the big focusing knob. It's called the coarse adjustment knob. And then the smaller focusing knob is called the fine adjustment knob. A couple of rules for using the scope. When you're using the scope, you always want to put the lowest power objective, which is the 4x one in most cases, over the slide. Start there. When you're trying to focus, at first use only the coarse adjustment knob to focus, and use it only on the low power objective. Once you can see the object that you're trying to look at through the scope, use the controls underneath the stage to move that object into the middle of your field of view. Make sure you center it. If you want to continue to blow it up more, you're then going to use the nose piece to rotate the 10x objective into place. For all objectives other than that lowest power objective, you only want to use the fine adjustment knob to focus. So you'll put the 10x objective into place, you'll use only this fine adjustment knob to focus it. If that's still not good, if you still need it to be bigger, you rotate the 40x objective into place. Again, only use the fine adjustment knob. The coarse adjustment knob should only be used on the lowest power objective. If you still can't get the object to focus the way you want to get it to focus, you can also go in and adjust the diaphragm, change the amount of light coming through your stage, and that will tend to clarify things for you. This type of scope, standard biology classroom scope, is called the compound light microscope. Something else that we want to talk about are the different kinds of cells that exist on Earth. And those two are mostly, the two that were famous are prokaryotic and eukaryotic. The word prokaryote means before the nucleus. Scientists think that the first living things to develop on Earth were bacteria-like organisms. Bacteria are unicellular prokaryotes. They're made of one prokaryotic cell. A prokaryotic cell does have some DNA, but it's just sort of floating around in the cytoplasm. There's no nuclear envelope around it. It doesn't have any membrane-bound organelles like Golgi bodies or mitochondria or chloroplast. It does have ribosomes because it needs to be able to make proteins. It has a cell membrane. It has a cell wall. It may have cilia or flagella to help it move. The other main type of cell is the eukaryotic cell. This word means true nucleus, or true kernel, as the actual meaning. But eukaryotic cells have DNA that's surrounded by a membrane. It's enclosed. It's like it's in a vault. They also have little packages or organelles inside them that have specific jobs. For example, the mitochondria is where cell respiration takes place and it makes energy. The ribosome makes proteins. The Golgi body right here actually helps to store materials. It's sort of like the cell's warehouse. It also may modify those materials while they're there. And eventually it's in charge of doing something called exocytosis, which is shipping things out of the cell. You might also think of it sort of like the UPS or the FedEx of the cell. Vesicles or vacuoles are storage containers. They may store water, food, waste. The rough ER transports materials through the cell. I compared it to sort of like a conveyor belt in a factory. It moves proteins through the cell. Also, while the proteins are in the rough ER, they're modified. And when they make it to the end of the conveyor belt, in this case the rough ER, they're packaged, usually in a vesicle, and they will be shipped to the Golgi apparatus and eventually out of the cell. Another organelle that we need to mention is the lysosome. It's not labeled on this diagram, but lysosomes have these powerful enzymes in them which help to break down things like old dead cells or pathogens or waste. And those, because of that, the lysosomes are sometimes called the recycling center of the cell. So the pieces of those things that are broken down are reused. This is an animal cell. Notice that there's no cell wall. The membrane is very flexible and fluid-like. Also notice there's no chloroplast. Animal cells obviously can't do photosynthesis. Here's a plant cell. Notice that a lot of the parts are similar to the animal cell. Plant cells are eukaryotic. They have a membrane-enclosed nucleus. Plant cells also have organelles. Here you see the Golgi apparatus, the mitochondria. In addition, they have chloroplasts because they can do photosynthesis and make their own food. Lots of organelles, mostly the same as the organelles in the animal cell. But one thing that you'll notice in the middle of most plant cells is this huge vacuole that's used to build up water pressure, store water, and it also helps to support the cell. So a couple of differences between plant and animal cells. Number one, the plant cell has chloroplast, the animal cell doesn't. Number two, the plant cell has this large vacuole in the center to store water. The animal cell has some vacuoles, but they're much smaller. And number three, the plant cell has a cell wall. It's made of a sugar called cellulose. Animal cells don't have a wall at all. So for the test tomorrow, make sure you know what each of the organelles does. Make sure you know some differences between plant and animal cells. Make sure you know some differences between prokaryotic and eukaryotic cells. And make sure you know the parts of a microscope and what they do. And also you know how to use the microscope. If you know those things, you will do very well on tomorrow's test. Good luck.